Hi friends, today we will discuss about another cellular organelle, that is endoplasmic reticulum. This video is beneficial to UG and PG students. So see, this endoplasmic reticulum is the most important cellular organelle. It is largest cellular organelle. If we see that the name of this endoplasmic reticulum, this endoplasmic reticulum is situated near the nucleus, and we know that the cytoplasm is divided into endoplasm, which is a part or a region of a cytoplasm which is near to the nucleus, and the ectoplasm, which is near to the plasma membrane. So the organelle which is present in the endoplasm, that's why the name is given as a endoplasmic. And reticulum means a net-like structure. So various kinds of tubules and cisternae are part of the endoplasmic reticulum. They form a net-like structure, and that's why it is called as a reticulum. This was first reported by Bruder in 1945, and endoplasmic reticulum is absent in egg and embryonic cells. Now, this endoplasmic reticulum is nothing but a kind of a compartment, a large compartment having a membrane-bound structure, and in that membrane-bound structure, it encloses a lumen, and this lumen contains a matrix, and this matrix is having a different types of enzymes, different types of proteins, and these are responsible for various kinds of modifications that are going on during the metabolism. So this protein modification is done with the help of this endoplasmic reticulum. Even a synthesis of a protein is also done with the help of endoplasmic reticulum, that we will see in a functions. So remember that this is a kind of a compartment, a spatial, it has a spatial function. So next is structure of a um, endoplasmic reticulum. See, you will find the endoplasmic reticulum is made up of three types of structures, um, or structural features, that is cisternae, vesicles, and tubules. Now, if we see the cisternae, that these are nothing but a sac-like structures, and these sac-like structures are long, flat, and they are arranged one above the other, and the membrane of this cisternae is in continuation with the outer nuclear membrane. So that means the perinuclear space is in continuation with the lumen of the endoplasmic reticulum, or we can say the endoplasmic reticular cisternae. Then another peculiar feature of endoplasmic reticulum is tubules. These tubules are nothing but the irregularly branched tube-like structures, and their diameter is about 50 to 100 nanometer. And these, we can say, the tubules, they are uh, actually uh, they are surrounded by the vesicles, small vesicles. These are also very small bubble-like vesicles, circular vesicles. So together they form the endoplasmic reticulum. That is cisternae, vesicles, and tubules. So they together form the endoplasmic reticulum. Now, this endoplasmic reticulum, the boundary wall of the endoplasmic reticulum is nothing but the plasma. So that's why we call it as a unity membrane. Why we call it as a unity membrane? That is, this particular type of plasma membrane, that means 50 to 60 nanometer plasma membrane, is present in the nucleus, it is present in endoplasmic reticulum, it is also present in a Golgi complex, it is also a part of a lysosome, it is also um, same in a plasma membrane also. That's why we call it as a unity membrane. So here also endoplasmic reticulum the boundary wall of this endoplasmic reticulum, or the limiting membrane, is nothing but the kind of a plasma. So this is all about the structure of endoplasmic reticulum. You can see here, this is the nucleus and this is the nuclear membrane. These are the pores of the nucleus, and you will find that the outer nuclear membrane is in continuation with the endoplasmic reticulum. So these are the flattened sac-like structures of endoplasmic reticulum. Here also you will find that these are sac-like structures, flattened sac-like structures, and along with that, small round-shaped bubble-like vesicles that are also present. So this is the overall structure of endoplasmic reticulum. Now there are two types of endoplasmic reticulum. First one is called as a smooth endoplasmic reticulum, and another is called as a rough endoplasmic reticulum. Now, why it is called as a smooth? That is, here you can find this in the smooth endoplasmic reticulum in the diagram. This smooth endoplasmic reticulum is a really smooth. It doesn't have any kind of ribosomes are attached to it. So its appearance is smooth, and that's why it is called as a smooth endoplasmic reticulum. While in case of a rough endoplasmic reticulum, you will find that the rough endoplasmic reticulum is having ribosomes attached over it, and that's why it appears like a rough, and that's why the name is given as a rough endoplasmic reticulum. Now, another peculiar feature of the smooth differences of in between the smooth and rough endoplasmic reticulum is that the rough endoplasmic reticulum have more or less flattened sacs are more in a rough endoplasmic reticulum as compared to smooth endoplasmic reticulum. So that is attributed to the kind of protein that is present in a smooth endoplasmic reticulum, that is known as reticulon. So that is the difference between the smooth endoplasmic reticulum and rough endoplasmic reticulum. Then functions of endoplasmic reticulum, we will see there are three actually the common functions are there, and functions of a smooth endoplasmic reticulum are different, and functions of a rough endoplasmic reticulum are also different. So that's why we have categorized it for the ease of understanding that it has a three types of, or we can say the functions can be divided into three types: common functions, functions of ACR, and functions of RER. So first of all, we will discuss the common to both endoplasmic reticulum. These functions are common to both endoplasmic reticulum. First one is it forms the skeletal framework. So naturally, you will find that the most of the endoplasmic part of the cytoplasm that is filled with the endoplasmic reticulum. So that's why it gives a kind of a cytoskeletal framework to the cell. So and because of that, it gives a kind of a mechanical strength to the nucleus as well as to the cell. So this is the first importance, or we can say the function. Then second function is active exchange and transport of a cellular materials. So ER membranes are responsible for exchange and transport of various materials within the compartment and outside the cell. So that is also a very important function of endoplasmic reticulum. Then third is the metabolic activities due to the presence of different enzymes. So different enzymes are present inside a endoplasmic reticulum. So whenever the proteins are synthesized in a endoplasmic reticulum, or lipids are synthesized in a endoplasmic reticulum, they are modified to a certain extent, and for that modification certain kinds of enzymes are required, and these enzymes are present in the lumen of the endoplasmic reticulum or on the membrane of the endoplasmic reticulum. So these kind of metabolic activities are performed by the endoplasmic reticulum. Then it provides increased surface area for a cellular reaction. So as the membrane of this endoplasmic reticulum is highly folded, it provides more surface area for um, various kinds of a cellular reaction. So that is natural. So then the fifth one is the formation of a nuclear membrane during the cell. And this is the most important aspect of functions of endoplasmic reticulum. That whenever during the mitosis or a meiosis, the nuclear membrane disappears, but the reappearance of the nuclear membrane is totally due to the endoplasmic reticulum. So these are the common functions of endoplasmic reticulum. Now we will discuss about the functions of a smooth endoplasmic reticulum. The first function is lipid synthesis. As you know that the eukaryotic membranes are made up of three different kinds of lipids: phospholipids, glycolipids, and cholesterol. Out of this, phospholipids are synthesized with the help of glycerol 3-phosphate and free fatty acids. Now, with the help of see, there is one enzyme, membrane-bound enzyme is present in the ER uh, membrane, and it has the active site towards the cytosol. So this membrane uh, converts free fatty acids and glycerol 3-phosphate into phosphatidic acid, and then this phosphatidic acid is incorporated in the membrane. Now, after incorporation, phosphate molecule is um, removed from the phosphatidic acid, and because of that, this phosphatidic acid is converted into diacylglycerol. Now, this diacylglycerol is a precursor for all the phospholipids that are present in the membrane. Remember that the all the phospholipids are synthesized in a ER membrane or in a ER except the sphingomyelin. So this is how that the different types of lipid molecules are synthesized in a smooth endoplasmic reticulum with the help of membrane-bound enzymes. So when these phospholipids are synthesized, you will find that these newly synthesized lipids are then incorporated in the membrane, and with the help of the flippase molecule, these newly incorporated lipid molecules are then transported to the other half of the ER membrane. So this is how there is a growth of both halves of the lipid bilayer. So this is about the lipid synthesis in a smooth endoplasmic reticulum. Now we will go to the second part, second function, that is glycogen synthesis. It has been also found that the smooth endoplasmic reticulum provides a necessary microenvironment in the hepatocytes. Now we know that what are the hepatocytes? Hepatocytes means the liver cells. So these enzymes, we can say the whatever the enzymes that are present in the hepatocytes, um, endoplasmic reticulum, and the substrate, they come in close contact, and where they interact with each other, and they brings about the synthesis of a glycogen. Now, what is the importance of this? It creates, or because of the foldings and um, infoldings of the smooth endoplasmic reticulum, smaller compartments are synthesized, and these smaller compartments are necessary, or they provide a kind of a microenvironment, and in that microenvironment, even a small concentration of endoplasmic reticulum will also suffice, and you will find that this particular enzymes and substrate, they will give rise to the product that is the glycogen here. So this glycogen synthesis is done in a smooth endoplasmic reticulum. Then third function of this is smooth endoplasmic reticulum is the steroid synthesis. For example, we know that different types of steroids are present: cholesterol, progesterone, testosterone. All these are uh, synthesized with the help of enzyme that is membrane-bound enzyme that is present in the endoplasmic reticulum. The name of that enzyme is the HMG coenzyme A reductase. Now, the C-terminal segment of this particular enzyme is located on the cytosolic side of the ER membrane, and it synthesizes the precursors of cholesterol. So, precursors of the enzymes, that is cholesterol, and these from this cholesterol, the different types of hormones are synthesized. For example, testosterone, estrogen, and cortisol. So, this is all about the third function of the um, smooth endoplasmic reticulum. Then the fourth function of the smooth endoplasmic reticulum is the metabolism of a carbohydrates. Now we know that whenever the glucose is available in the blood, the from the blood the glucose is taken to the liver, and in a liver cell this glucose comes in a hepatocytes cytoplasm. The cytosolic, we can say the cytosolic glucose is called as a glucose 6 phosphate, or it is converted into glucose 6 phosphate. So that whenever a glucose comes into the cell, it is converted into glucose 6 phosphate, so that the concentration of glucose remains low inside the cell or the hepatocyte, and so that there should be a constant flow of the glucose from the blood to the hepatocytes. So when this glucose 6 phosphate is converted, this glucose 6 phosphate is taken to the endoplasmic reticulum with the help of another membrane bound enzyme that is known as a, or sorry, not enzyme, it is a transporter that is known as a glucose 6 phosphate transporter. And this G6P then taken into the ER lumen, where again a membrane bound enzyme is there that is known as a G6 phosphatase, glucose 6 phosphatase. It converts, or it removes that phosphate molecule from the glucose, from the glucose 6 phosphate, and converts into glucose and phosphate molecule. Again, the different transporters are there for taking up glucose, or taking back that glucose from endoplasmic reticular lumen to the cytosol. So, in all, it maintains the glucose homeostasis. So, this is the role of endoplasmic reticulum with the help of an enzyme that is G6 phosphatase. This glucose metabolism is uh, performed or maintained. We can say the glucose homeostasis is maintained by with the help of this smooth endoplasmic reticulum. Then fifth function of the this smooth endoplasmic reticulum is that detoxification. So this is the most important function of the uh, endoplasmic reticulum. We know that there are different types of drugs are the poisonous material, or we can say, for example, the alcohol. Alcohol is a kind of a poison. So the drugs and the alcohol, when they are ingested in our body, they are taken to the liver, and in a liver they are processed into a water soluble form, and then they are discarded out of the body. So this kind of a function is mediated through a smooth endoplasmic reticulum. There are two enzymes that are present in the smooth endoplasmic reticulum. That is, one is a cytochrome 450 system, and another is a UDP glucuronoside transferase. These enzymes, they are responsible for the conversion of this particular uh, poisonous material, uh, uh, poisonous material into a soluble material, water soluble material, and then it is taken to the bile or to the urine, and so that it should be it can be expelled out of the body. So This cytochrome 450 system is present in the smooth endoplasmic reticulum, and the UDP glucuronosyl transferase is also present in the endoplasmic reticulum that helps in detoxification of potentially hazardous drugs like alcohol or any other type of drugs. So this is the detoxification. The last sixth function of the smooth endoplasmic reticulum is it stores the calcium. So the large store of calcium is maintained by the smooth endoplasmic reticulum, and this storage of calcium that is useful in signaling uh, various kinds of that we will see that is useful in a signaling. So it acts as a reservoir for the calcium. So in case of a sarcoplasmic reticulum, you must have heard about this. This is a smooth endoplasmic reticulum, and it stores the calcium in a muscle cell. So this is about the functions of smooth endoplasmic reticulum. So thank you very much for patient hearing. Please subscribe to my YouTube channel, BAMU'S Biogenius, for latest uploads, or like my videos and comment. If you have any queries, you can ask me on the comment section. So thank you very much once again for patient hearing.